Hi, my name is Elise Docena and I go to Washington Liberty High School in Arlington, Virginia. My project was the effect of the melting of Arctic ice on phytoplankton abundance. Recently, Arctic ice has been melting, which releases fresh water into the oceans. This takes an effect on our currents, specifically the rotation and conditions within. I chose to study phytoplankton because they play an important role on the Earth. My experimental question was, does the melting of Arctic ice affect phytoplankton abundance? There's actually been a previous study that found a change in the dynamics of the phytoplankton community in the Beaver Gyre caused by Arctic ice melt. However, they had no data on whether or not phytoplankton abundance as a whole increased or decreased. It's important to monitor how climate change affects the animals, especially those such as phytoplankton that influence their ecosystem in a great way. My hypothesis was, if the average Arctic ice area decreases throughout the years, then the phytoplankton abundance will decrease as well, indicating a positive correlation. This was implied due to a decrease in nutrient stocks in the Beaver Gyre resulting from Arctic ice melt, nutrient stocks being the thing that phytoplankton rely on to live. Data was collected purely from online databases. The independent variable was Northern Hemisphere average Arctic ice area in millions of kilometers squared, and the dependent variable was chlorophyll concentration in milligrams per meters cubed. Data for the independent and dependent variables was taken from August of 4 different years, and the data for the dependent variable was also taken from 8 different coordinates spanning the Beaver Gyre. Chlorophyll concentration actually correlates with phytoplankton presence since phytoplankton carry out photosynthesis. I had to take extra steps to retrieve the numerical values for chlorophyll concentration in the water, as the chlorophyll concentration dataset was displayed as a color-coded map. Different colors on the map indicated different chlorophyll concentration levels, which you could find by comparing the color of the section on the map to the color bar below, which was labeled with correlating numerical values. On the left is pictured the setup for the dataset for chlorophyll concentration with the map, and on the right shows me comparing two screenshots, one of the area on the map and one of the color bar, and overlapping them so I could find out exactly which color was being displayed on the map. On the top is my raw data table, which shows you the explicit chlorophyll concentration levels for each of the 8 different coordinates for each of the 4 years. And below that we have the summary data table, which just shows the average chlorophyll concentration over the whole area and the standard deviation. At the bottom left, we have a bar graph depicting average chlorophyll concentration at different average Northern Hemisphere Arctic ice areas. And the orange bar just shows where the average was, and the line above that shows the standard deviation. On the bottom right, we have average chlorophyll concentration over time as a line graph. At the top, we have a scatter plot with a line of best fit, which shows you that there was a positive correlation between Arctic ice area and chlorophyll concentration. And on the top right, we have a comparison line graph of the changes in both Arctic ice area and chlorophyll concentration over time. The structures on the line graph differed greatly over time, which indicates that the changes in chlorophyll concentration did not take place solely because of the changes in Arctic ice area. Otherwise, the lines would have steepened at the same points and taken on the same overall shape. So while chlorophyll concentration did decrease alongside Arctic ice, with evidence towards a strong positive correlation as well as I predicted, the null hypothesis was still accepted. There was no significant statistical difference found between the chlorophyll concentrations at the different Arctic ice areas. The standard deviation for each trial also varied a lot. The highest value even reached 6.57. This is somewhat expected since the measurements for chlorophyll concentration were taken from different coordinates spanning the whole gyre. My experimental hypothesis was rejected as the null hypothesis was accepted. One possible explanation is that, as mentioned before, there was a study that showed that certain types of phytoplankton became more abundant in the gyre while others decreased due to the influx of fresh water. So perhaps some types of phytoplankton thrive in the conditions created by the melting of Arctic ice, whereas others decline, which would ultimately cause little to no change in the overall abundance of all the types of phytoplankton. Additionally, the data may have been skewed due to the placement of coordinates. Not all the coordinates may have shared the same environmental conditions. Coastlines are actually a better habitat compared to open ocean. So if this project were to be redone, I would move the data retrieval locations higher north, further away from the coastlines. And additionally, I would change the distance between the data retrieval points to be smaller. And if these changes are made, then the entirety of the data would be under the same or extremely similar environmental conditions, allowing for a more reliable result due to precise data. Thanks for watching.